Evaluation of inflammatory response and DENV replication in platelet-monocyte interaction model. I'm Cecíllia, I'm a PhD student, and my advisor is Professor Eugênio Hottz. Dengue is caused by dengue virus, which belongs to the genus Flavivirus, and is comprised of 4 antigenically distinct serotypes. DENV is composed of a single-stranded positive sense RNA that is translated into a polyprotein. This polyprotein is cleaved into the structural proteins, C from capsid, E from envelope, and M from membrane, and 7 non-structural proteins. NS1 is one of the non-structural proteins, and along with the other non-structural proteins, it forms the replication complex, which is responsible for the synthesis of new RNA molecules. NS1 is highly conserved amongst flavivirus, and it can be either expressed on the cell surface as a dimer or secreted into the bloodstream as an hexamer. Dengue is transmitted mostly by Aedes aegypti mosquitoes, and around 390 million new cases are registered every year. Dengue infection can be asymptomatic, mild, or evolve to severe dengue. Severe dengue is caused by high release of inflammatory mediators, which are related to vascular leakage. Furthermore, thrombocytopenia is one of the most studied characteristics, and it can be found either in mild and severe dengue. However, the lowest count of platelets are found during severe dengue. Even though thrombocytopenia has already been well characterized, it is not known whether platelets are capable of replicating dengue. Simon and colleagues have suggested that platelets sustain a productive replication, while Carr et al. Have suggested that platelets lead to an abortive replication. So, more studies are necessary to understand how far platelets sustain dengue replication. Platelets were first described as mediators of the coagulation system. However, platelets are now recognized as cells of the immune system. This can be explained by the expression of receptors on the platelet surface, such as TOL4, that are capable of recognizing dumps and pumps. Moreover, platelets store inflammatory mediators, such as RUNS and PF4, and also adhesion molecules, such as P-selectin. Platelets also possess stored RNA that can be translated into proteins. Besides, platelets can interact with leukocytes, leading to migration of such cells to the site of injury and inducing cytokine secretion and its formation. Platelets are also able to recognize several types of pumps and dumps, leading to aggregation, granule secretion, adhesion, and several other responses. Although platelet infection hasn't been fully elucidated, it is well established that platelets can contribute to immune response during dengue infection. A study from our group has shown that platelets from dengue-infected patients are able to induce the secretion of cytokines by monocytes in an ex vivo model. Platelets themselves are capable of releasing inflammatory mediators. However, contribution of dengue-infected platelets to monocyte activation, leading to cytokine secretion, requires further studies. High levels of NS1 are found on the plasma of severe dengue patients. It has already been demonstrated that NS1 binds to TOL4 on the surface of endothelial cells, damaging the vascular integrity and causing vascular leakage. Moreover, NS1 interacts with TOL4 that is expressed by immune cells and it leads to the release of pro-inflammatory cytokines, which act upon the endothelium and increase the damage. Recently, it has been shown that NS1 is also capable of interacting with TOL4 expressed on platelet surface, leading to thrombotic and hemorrhagic response. However, it is not clear whether NS1 induces a platelet inflammatory response. Hence, we aim to investigate the contribution of dengue-infected platelets in monocyte inflammatory response during dengue infection, and also evaluate whether platelets contribute to virus replication in monocytes. I would like to thank CAPES, Federal University of Juiz de Fora, and the Biological Science Graduation Program for the financial support. I would also like to thank all our colleagues and professors of the Biochemistry Department, LEAP, Cell Biology and Immunology Labs, and also our colleagues from Fiocruz and the Federal University of Rio de Janeiro. Finally, I would like to thank my advisor, Professor Eugênio Hottz. Thank you.